This lecture concerns proteins, how the DNA genetic code is used to literally teach the cell how to assemble the amino acids into the thousands of proteins that do the work required to keep it active and alive. This process, called protein synthesis, is the one process that overrides all others, since it is the proteins that it produces that actually become part of almost every cell part and actually operate the biochemistry of the cell. No life would be possible without the proteins made by this overwhelmingly powerful process. Biologists call protein synthesis the central dogma of molecular biology, from which all other activities are made possible. In this lecture, we will cover how a gene's worth of DNA is read like a book, and how the information is used to assemble amino acids in the proper order and length to make a fully operational protein. Now, let's take a look at the structure of a DNA molecule as a quick review. We know that DNA has two sides. It's built like a sort of a spiral staircase, and if you straighten it out, it looks kind of like a ladder. The two uprights of the ladder, or backbones of the DNA, are made of deoxyribose sugar, which is symbolized by this pentagon here with D inside, and phosphate. And they just alternate and alternate and alternate, and it just keeps going literally for thousands long. DNA is a very, very big molecule. On the other side, you have another backbone that's sort of oriented in the opposite way. Inside of the DNA is the code, and the code is made up of these four nitrogenous bases. The bases are called adenine, thymine, cytosine, and guanine. Whenever you have an adenine on the DNA molecule, it must match with a thymine on the other side. They fit together like a hand and a glove. Cytosine on one side always matches with guanine on the other. So A always goes with T and C always goes with G. That is the structure of DNA. Now, if I was to circle this amount of DNA here, it consists of one of everything: one deoxyribose, one nitrogenous base, in this case adenine, and one phosphate. That structure there, that's called a nucleotide, and the nucleotides are the monomers that the DNA uses, what the cell uses to make the DNA, to build a DNA. It links them all together when it needs to make a DNA. This is a cytosine nucleotide here. It's one phosphate, one deoxyribose, and one carbon. In this case, I'm sorry, one cytosine. It's the cell's program. Now, RNA is a little different. RNA is only one side, and it has ribose sugar instead of deoxyribose, and it has the guanine and the cytosine and the adenine. But in place of thymine, it has a uracil nucleotide, or a uracil nitrogenous base. This is a uracil nucleotide right here. It consists of one phosphate, one ribose, and one uracil. This is a cytosine nucleotide. This is an adenine nucleotide. And you can see how the cell puts this together, nucleotide by nucleotide by nucleotide. RNA is DNA's messenger. DNA is too big to get out of the nucleus, so it can't really get to where the proteins are made in the cytoplasm. So it has to send out a small messenger with the information on it. That messenger is called messenger RNA. Now, this is an amazing picture of a tobacco plant. That tobacco plant is actually glowing using the proteins that a firefly makes in order to make it glow at night when you see fireflies flying around outside in the dark. What they did is they inserted genes into the DNA of the tobacco plant, and it was the genes they took from the firefly. The tobacco is actually using the genes from the tobacco plant and using exactly the same code on the DNA that the firefly uses, is actually making the same protein that the firefly makes. And that protein allows the plant to literally glow in the dark. This is an animal gene making animal proteins in a plant. The genetic code on the DNA for making specific proteins is the same for all animals and plants. This plant is using the same code on the DNA to make the protein as the firefly did. Now, the genetic code is like a language. This is very important. It has letters that make up meaningful words, just like a language does. The genetic language, or code's letters, are the 4 bases within the DNA: the adenine, the thymine, the guanine, and the cytosine, A, T, G, and C. On the RNA, it's A, U, G, and C, because remember, on RNA, we substitute a U for a T. Each genetic word now, this is the important part. A language is not just letters. Letters become important when they become words. Each genetic word is 3 letters long, or 3 bases long. There are 64 combinations of 4 letters, taking 3 at a time. Now, the 4 letters are A, T, G, and C on the DNA, and if you take them 3 at a time to make words out of them, you can make 64 different combinations. There are therefore 64 words in the entire language. Each one of those 3-letter words is called a codon. Each codon stands for, or codes for, a specific amino acid. For example, the sequence AUG on an RNA molecule only stands for the amino acid methionine and no other. So whenever that codon appears on the RNA messenger, methionine is placed into the amino acid chain. AUG is literally translated as methionine. Remember, there are 20 different amino acids, and each one has a 3-letter code that would be translated into it. The order of the codons, or genetic words, can therefore determine the order of the amino acids in the protein that the gene is making. Now, this is the actual genetic code. In 1968, you can see these scientists here actually won the Nobel Prize for breaking the code. You notice that these are the codons here. They're all 3-letter bases long. This one says adenine, adenine, uracil. These are codons on the messenger RNAs. So these are RNA codons. AAC is adenine, adenine, cytosine, and so on and so on and so on. You can make 64 combinations of 3-letter. When you have 4 letters, to taking them 3 at a time. Now, what's very important here is that whenever there's an AAU on the messenger RNA, that cell is going to read that and put asparagine amino acid into the protein. Whenever there's an AGG, it will read that and put arginine in the protein. This is the way the code is actually done in all life forms on the earth. In other words, ACC means threonine. In human beings, when we have an ACC on our messenger RNA, the cell will read that and put a threonine into the protein it's making. If it was an oak tree, an ACC on its messenger RNA, guess what? Would put a threonine in the protein it's making. The genetic code is universal throughout life on the earth. And these are all the 20 amino acids. You're never going to have to memorize their names or anything like that. Just understand that each 3-letter code stands for an amino acid. It can be translated into amino acid, just like Spanish can be translated into English. One word means another. In this case, CAA means glutamine. Now, how does it work? Here is a DNA molecule in black. And you see this red structure here. This is an enzyme. The enzyme is called DNA polymerase. And what it's doing, it's moving down the DNA strand and it's opening it up like a zipper. It's actually spreading apart the 2 sides of the DNA. This side here is known as the coding strand. That's the one that's going to be the genetic code. This one on this side is just going to be sort of like the cover of a book. When this is all done, that'll close back up again. Now remember, T always goes with A, and A goes with U if you're making an RNA. This blue thing here is messenger RNA. This is an RNA molecule that's being produced inside the DNA using this side of the DNA as a guide. This is a gene. We have inherited this order of bases from our parents. When you make the messenger RNA, because remember, the DNA can't get out of the nucleus, it's much too big. It has to send one gene's worth, which is enough DNA to make one protein, of RNA out into the cytoplasm to make the protein. So when you're making the messenger RNA, you produce it. This RNA polymerase actually starts to make the RNA molecule as well as opening up the DNA. It puts an A where there's a T. Now remember, A would you normally go with a T, but this is RNA you're making, so you'll put a U there instead. C goes with G, C goes with G, G goes with C, T goes with A goes with U, and so on. It'll just keep going until it reaches the end of the gene. Now, there could be hundreds and hundreds of genes on one chromosome. This is just going to make a messenger RNA that's the length of just one gene, which would be enough information to teach the cell how to just make one protein. If there's hundreds of genes on the chromosome, that chromosome has the ability to make hundreds of proteins, depending on the codes. Now, that messenger RNA is going to peel off of here when it's finished, and it's going to come out into the nucleus out here. This is the nucleus of the cell here. Inside the nucleus, the RNA is going to be modified. The RNA actually consists, messenger RNA consists of what they call exons and introns. Introns are nonsense pieces, garbage, probably caused by virus DNA that has been inserted into our DNA or the host DNA over the centuries. This has to be cut out. So this is nonsense RNA. It's from nonsense gene sections of the chromosome. There are things in the nucleus called spliceosomes. What they're going to do is they're going to attach to the messenger RNA and literally cut off the introns, let them out. These are garbage now. They're going to come out and then link together the exons. The exons are the good strands that actually have the code that you need to make the protein. And here it is, the actual messenger RNA molecule that has been produced after you have taken out the garbage. And this happens inside of the nucleus. Now, once this is done, here is a messenger RNA here. These are the introns. You got to get them out of there. Here's another intron. You got to get it out of there. The spliceosomes are going to cut these guys out and splice together the exons. And when that happens, you end up with a complete messenger RNA, which has the right genetic code on it, taking out the nonsense material that was in the DNA from viruses in the past, when the viruses attached their DNA onto that person or animal or plant's DNA in the past. That virus DNA has to be removed. And that is the introns here on the messenger RNA. Another thing they do is they put a cap on either end, and the caps are very, very different. And the reason for that is so that the cell knows where to start reading the code and where to end reading the code. You don't want to read a sentence backwards. So you have a beginning here and you have an end to the messenger RNA. Now, when the messenger RNA comes out of the nucleus, it goes into a ribosome. It actually attaches onto a ribosome. Now, the process of making that messenger RNA in the nucleus was called transcription. And the word actually just means to copy. You're basically making a copy, sort of a backwards copy of the gene as messenger RNA. This stuff here that you see in what's going on in the ribosome is called translation. This is how we translate the genetic code into the language of proteins. The ribosome has the messenger RNA attached to it. And you can see the messenger RNA has a code on it that it got from the DNA. And in the ribosome, there are sort of niches or dents. And here you have AUG, the very first codon. Now, you know that A always goes with U, U always goes with A, and G always goes with C. They join together like little magnets. There's another kind of RNA in the cell That other kind is very, very important. It's called transfer RNA or tRNA. Transfer RNA has what they call an anticodon on one end of it and a specific amino acid, and only that kind, one kind of amino acid on the other end. So any transfer RNA that has a UAC anticodon is always going to carry methionine on the other side. UAC transfer RNA will not carry any other amino acid. Now, UAC only matches AUG. It will not link on any other codon on the messenger RNA. A has to go with U has to go with A, and C has to go with G. These will magnetically attach here and bring with it its methionine. So AUG here is being translated as the amino acid methionine. In the second notch, you have the second codon on the messenger RNA, GCA, and GCA will be attracted to an transfer RNA that has its opposite codon, its anticodon, and that would be G going with C, C going with G, A going with U. Now, a CGU anticodon transfer RNA will only carry the amino acid alanine. So that means that if anytime there's a GCA on the messenger RNA, this transfer RNA is the only one that's going to attach to it, and it's going to bring to the story only the amino acid alanine. Now, once these two guys are near each other on the ribosome, they will join together, they will bond. And what happens here, you're starting to link amino acids together in the order that these codons are telling them to do. That is the beginning of making a protein. Now, if you go over here a couple of minutes later, you see what's happened. The messenger RNA is moved over one codon, and now the third codon is in the second notch here, and that's UCG. The anticodon that matches that is AGC. That's going to stick on there, and that one's only going to carry serine. And what's going to happen here is that the AGC transfer RNA that only carries serine will put it in this location as it attaches to the UCG codon. And then the serine and the alanine will join together to make the next amino acid in the protein. After that, the transfer RNA will move over another notch. The next codon here will end up being here in the notch. The next transfer RNA will attach to it, put the next amino acid in, and keep going and going and going and going and going until the protein is complete. And that would be, literally hundreds and hundreds of amino acids long. Once it's done, the amino acid chain comes off of the ribosome and starts to become processed in the cell to be an operational protein. Now, the transfer RNA that loses its amino acid, like right here, this one, that has come off. That was this one over here at one time. It lost its methionine. That's going to go back into the nucleus and pick up another methionine, or even in the cytoplasm, it'll pick up another methionine and be used again to make another protein. This is a transfer RNA, and you can see here, this is where the amino acid attaches to it. And there's the anticodon right there. And this one is AAG, and a specific amino acid would attach to this end. So you remember the transfer RNA has a specific amino acid on one side and a anticodon on the other side. The anticodon would match the codon on the transfer RNA, on the messenger RNA. And if the messenger RNA had UUC, this is the one that would attach to it and would bring its amino acid to the story. Now, this is an amazing photograph of the process actually happening for real. These are a series of ribosomes here. These are all a bunch of ribosomes, all these dots. And if you look carefully, you'll see there's a sort of a strand that is kind of going through all of them. That's the messenger RNA. The messenger RNA is moving through these like almost like a little train going by. And as it goes by, each one of these ribosomes is producing a chain of amino acids. And you see this is the protein starting to be made. Each ribosome is making the many copies, or actually there's many copies of the protein being made from this messenger RNA because there's many ribosomes doing it. Once the protein, the amino acid chain is put together, then what happens is this is an amino acid chain. This is what comes off the ribosome: leucine, lysine, histidine, lysine, valine, leucine, glycine, alanine, valine. These are the amino acid in the order according to the code that was on the DNA and the messenger RNA. What it does is, this is the primary structure. It's just a chain of amino acids. It doesn't work in the cell. It needs to become a shape. And so what happens is that it starts to coil up. And you can see I'm trying to show that it's made into a coil here. And that when it does that, it becomes sort of like a spring of amino acids. And that's called a secondary structure. And then what happens after that is that the string of this sort of spring of amino acids, it just kind of tangles itself up. And just it's like a slinky that you just tangle all up inside. And it becomes a specific shape. Once it becomes a shape, then it becomes active in the cell. Now, some amino acids contain sulfur, and there would be one right here and one right here. And what happens is that where the amino acids that contain sulfur exist in the chain, they form disulfide bonds that causes the way this thing folds up. So the location of the sulfur-containing amino acids really determines what shape this thing is going to end up being. This could be an enzyme. It could be a part of a fingernail. It could be all sorts of different types of proteins. It could be hemoglobin, carrying oxygen around. It could be a hormone. It could be a pump that pumps things in and out of the cell. It could be an enzyme. All of these things are what proteins do. They're very important. Sometimes a protein isn't finished until it links to another protein. And here we have one tertiary structure here and another one here linking together, like right there, with these two sulfur bonds again. This is called a quaternary structure, or fourth degree. And an example of one of these that have actually two amino acid chains made by two different genes that link together eventually in the cell. An example of that would be insulin. Insulin has what they call an alpha chain and a beta chain. And once it's put together, then the insulin works to help control the sugar in your body. Now here, what happens is interesting one here. What happens if there's a mistake, either in the DNA or the messenger RNA? And let's say a CTT on the DNA is where it's supposed to be to make the protein hemoglobin. That would produce a messenger RNA in this spot with a GAA codon. That will put eventually the amino acid glutamine in hemoglobin. GAA is the code for glutamine. But what if that was an A there instead of a T in the DNA? It was a mistake. When the DNA copied itself, instead of putting a T where it belongs here, it puts an A instead. And what will that do? It'll make a messenger RNA with the wrong codon. This is the codon it's supposed to be, GAA. But this is CAT, so it's going to make GUA. And that's going to put valine in there. So what's going to happen is that the hemoglobin is going to have one wrong amino acid in it. And that is the cause of a disease called sickle cell anemia, where many people suffer drastic problems with their red blood cells because of it. And it's all caused by a mutation. A mutation is a mistake. When the DNA has the wrong base in it, in the spot where the T is supposed to be, there's an A instead. You put the wrong amino acid in the protein doesn't work right, and the hemoglobin doesn't carry the oxygen or make the cell work the way it's supposed to. And your red blood cells become the wrong shape and they don't work very well. Now, as far as enzymes go, enzymes are usually tertiary proteins. That means that they are folded up into a certain shape, and the shape has sort of these little notches in it. And let's say this is an enzyme or a protein that is the one that's going to split, let's say, a disaccharide into two monosaccharides. So let's say this is the disaccharide sucrose. If that sucrose that means this is going to be fructose and that's going to be glucose linked together. It's a double sugar. We need to digest that in order to use it in our cells. We don't use the sugars as double sugars. We have to break them down into single monosaccharides. So what happens is that these notches on the enzyme, because of the way the protein folded up, fit exactly onto the sucrose. And here is the sucrose fitting into that active site on the enzyme. Now, when it's in there, it kind of doesn't fit exactly right. And so this one fits in here and this one fits in here. And that sort of stresses the bond between the two sugars. And eventually that bond's going to break. And when it does, the two sugars come off as single monosaccharides, and we can use those as food. This is the process that we use to digest our sugar and other foods that we eat. We break them down by attaching them to proteins called enzymes, and the enzymes will cause them to break their bonds and become smaller molecules that we can use for food. Okay. Now here you have an interesting one. Sometimes the product, now this is one of the products here, is fructose. That could land on a different site of the enzyme. And when it does that, it kind of changes the shape of the active site. So now the enzyme is shut off. So this is a sort of a test to see if you make too much fructose or too much product, the product can actually feed back and shut off its own production, so you don't make any more until you need it later. It's kind of like the heat in your house. You know, if it gets really warm, the heat itself is the product of your furnace. It will shut off the furnace if it gets too warm in the house, and it causes the thermostat to shut the furnace off so it doesn't make any more heat. So this is a control mechanism to keep making sure that when you break down something like this, you don't do it too much. And so what happens is that one of the products will feed back and land on this thing called an allosteric or other site on the enzyme, changing the shape of the active site. And so temporarily the enzyme is inactive, and you can't make any more sugar if you have enough. If you can't make any more monosaccharide sugar if you have too much of it in the cell. And this is just the last slide showing again how enzymes work. Here's sucrose again, landing on the active site of an enzyme. It breaks the bond, and you have two monosaccharides, fructose and glucose. But the interesting thing about that is once they come off the enzyme is right here, and it can be used again. So a small number of enzyme molecules can be used over and over and over again to keep digesting your sucrose. So that's the lecture on proteins and protein synthesis. And there's reading for it, and I hope that you got most of that information clear. Okay.